So the TATTON study is a multi-arm phase 1 trial taking one drug, AZD9291, which is a new EGFR inhibitor that's quite tolerable, and combining it in an intelligent fashion with 3 other targeted therapies in multiple phase 1 trials. So a trial that adds a MET inhibitor, savolitinib, a MEK inhibitor, selumetinib, and a PD-L1 inhibitor, the MEDI compound from MedImmune. And these 3 phase 1 trials each are targeting patients with EGFR mutations who failed prior EGFR TKI. A patient presented having acquired resistance, and they would then be randomly assigned to one of these different combination studies to test the tolerability of such intelligent combination therapies. Prior EGFR inhibitors like erlotinib or afatinib have more toxicity, especially more skin and diarrhea toxicity, and therefore are more difficult to combine. So the question is, now that we have this new EGFR inhibitor that's quite tolerable, will it be easier to combine it with other intelligent targeted therapies? And so the MET arm was intending to see if this can then be used to target MET resistance. The MEK arm with selumetinib was aiming to study eventually resistance based on ERK amplification, which has been studied preclinically, and then the PD-L1 arm, because these PD-L1 inhibitors are so exciting, hoping to combine these in a tolerable way. What we found is that each combination was tolerable. They were able to dose escalate to the full dose of each drug, and there were some toxicities. The primary toxicities were seen in the selumetinib arm with some diarrhea and nausea. 2 different schedules of selumetinib were studied, a continuous daily dosing or an intermittent dosing, 4 days on, 3 days off. And they ended up selecting the intermittent dosing of selumetinib to take forward, which is more tolerable, daily dosing of the savolitinib MET inhibitor at 600 milligrams daily, and full dose of the MEDI compound. These 3 combinations, now that we have a dose, are going to go forward. The next question is, where do we take these combinations now that we know we can give them and give them at a tolerable dose? So the MET combination is going to be going into MET positive resistance to EGFR inhibitors, both in the second line after failure of a drug like erlotinib or afatinib, and in the third line after failure of a drug like AZD9291 or ruxolitinib. There'll be an arm targeting MET resistance where patients with resistance to 9291 then could go on one of these combinations. The MEK combination will be in the third line, trying to take patients again who have resistance to AZD9291, adding the selumetinib to see if you can regain sensitivity. And finally, the MEDI compound, that combination, which is very exciting to patients because they want to get the EGFR inhibitor and the PD-1 inhibitor together. That's going to be studied in a first-line expansion cohort to see if patients are interested in getting these 2 potentially synergistic compounds together up front. And there also will be a separate trial run by AstraZeneca looking at patients T790M positive, like were enrolled in the AURA trial of AZD9291, randomizing them to the T790M inhibitor or the T790M inhibitor with the MEDI compound, the PD-L1 inhibitor, seeing if immunotherapy adds to the EGFR inhibitor. So it's a very complicated trial. The main lesson we learned is this is an efficient and effective way of getting combinations developed quickly. And now what we need to do is find a place for these combinations and figure out who should get them. And that's biomarker research to find the right combinations. I think the most exciting result I saw was that in one patient who had MET-dependent resistance but did not have T790M, a patient that I cared for, she had received prior erlotinib and afatinib with no benefit. She started AZD9291 with savolitinib and has had a dramatic and durable response, making us think that this is now a direction into MET-dependent resistance that could be very effective. This trial is really focused on EGFR-mutant lung cancer, which is maybe 15% of lung cancer. And that's a space where we're making a lot of headway and we're trying to make more headway. The fact is we have drugs, but they develop resistance. And at a certain point, these patients drop off the targeted therapies, start chemotherapy, and they have a harder time and have more toxicity. So what we're trying to do is do better for those EGFR-mutant patients by smarter combination therapies. EGFR-mutant lung cancer, these people keep coming back to our clinic, right? And they're living longer, they're on pill therapies, they're feeling well. They want targeted therapies, they want to avoid chemo. That's really the space where we're going. Every clinic around the world has these patients on erlotinib, gefitinib, afatinib, doing well and looking for another exciting targeted therapy option instead of chemo. That's what we're trying to focus on.